We have the honor to have Professor Andy Pekosz to give this talk. Dr. Pekosz is the professor and vice chairman of Department of Microbiology and Immunology in Johns Hopkins University. He has his Ph.D. at University of Pennsylvania in 1997, and he's the fellow of American Academy of Microbiology. He was the president of American Virology Society of 2018. He has won many awards. For example, Shikani El Hebrew Prize for Discovery and Innovation, Diversity Leadership Award, etc. His main research interest is to understand the interactions of viruses with the respiratory epithelium, especially focused on SARS-CoV-2 and influenza viruses. His laboratory teamed with many international scientists and made many important and significant discoveries in COVID-19 infections. Well, I'm really glad Dr. Pekosz will join us, and the speech of his talk will be the role of laboratory-based basic research in the clinical and the public health response to COVID-19. Well, ladies and gentlemen, let's welcome Dr. Andy Pekosz. Andy, please. Yeah.
Thank you very much for that kind introduction, and thank you to the organizers for inviting me here. I've had the pleasure of visiting Taiwan on a couple of occasions, enjoyed not only the science, but also the people and the culture. And I really hope that in the near future, we'll be able to get together in person to have some more fascinating scientific sessions like this one. So today I wanted to talk a little bit about the role of laboratory-based basic research in the clinical and public health response to COVID-19. It's quite obvious how clinicians and epidemiologists and public health officials can play a role in a pandemic. But I wanted to focus a little bit on what basic scientists can do to contribute to our knowledge that has a direct impact on how public health officials can respond to a pandemic. I'm going to use a few examples from my own laboratory's research. Obviously, there have been dozens of laboratories around the world that have done similar things to help bring information to light that will help respond to the pandemic. But again, I'll focus on the work that my laboratory has done and its implications on the direct response to the SARS-CoV-2 pandemic. So first, I need to acknowledge the members of my laboratory shown here. Fully half of these individuals have joined my laboratory since the pandemic has started. So I really give them a lot of credit in terms of being able to adapt to working in a new laboratory with a new professor and doing all of this in the middle of a pandemic and all of the constraints that the pandemic has put on us, particularly here in the U.S., but obviously globally. I'd also want to acknowledge here the large group of both funding agencies as well as centers that have contributed to my laboratory's COVID-19 response. When it comes to dedicating funds, dedicating effort, dedicating resources to the pandemic, a number of agencies have stepped up to really help us respond and do our work efficiently so that we don't have to worry about how we pay for things, how many people we have, how much laboratory space we had. All of our needs were met by our institution as well as other funding agencies to allow us to really focus on the work and the science that we have to do. So our response to COVID-19 actually starts with another important respiratory virus, and that's influenza. And together with my co-director, Dr. Richard Rothman, who's here at the School of Medicine at Johns Hopkins University, we established the Johns Hopkins Center of Excellence in Influenza Research and Surveillance, JHCERS is our acronym, about 8 years ago now. And our task was really to use the seasonal appearance of influenza as a means of setting up a pipeline to analyze both the viral genetic mutations that occur in influenza on an annual basis, but to also incorporate them into a larger picture of host responses to infection and bring all of that information together to help better our response to seasonal influenza, with the idea that our improvement of responses to seasonal influenza would reflect in our better ability to respond to a influenza pandemic. So what we do is we use situational awareness. We monitor for influenza-like illness and various other factors that would indicate in a non-specific way that there's an increase in respiratory illnesses in a population. We then go to broad-scale testing and rapid diagnostics so we can quickly identify what is the pathogen that is causing this increase in influenza-like illness in a particular geographic region. We then use sequencing technologies and other genetic characterizations to get whole genome virus sequences from those virus strains, and then we use a variety of bioinformatics approaches to try to triage and select particular isolates which we believe have mutations which are particularly relevant to that virus's spread in the community in that year. We then take those viruses, we either make virus isolates or we generate plasmid DNAs where we can generate recombinant viruses, and we study those in the laboratory to look at the general properties of virus replication, drug resistance, and host responses to infection. The individuals that provide the viruses that we study also provide serum and plasma, which we can analyze for the antibody responses generated within that individual and how they cross-react to other strains that are circulating. And again, all of this is thought to move into the public health response so that we can improve our response to respiratory virus infections. Our work is actually done at 3 different sites around the world. We have our primary site at the U.S. Capital Region at Johns Hopkins Medical Institution, where we have a racially diverse urban population. We have influenza vaccination, and this represents the Northern Hemisphere or seasonal influenza. We actually also have a very long-standing and great collaboration with Changong Memorial Hospital in Taiwan, which represents our surveillance for the Taiwan Capital Region. And as I'm sure you all know, Taiwan is an Asian urban population with influenza vaccination. It fits the Northern Hemisphere in terms of spread of influenza and the timing of influenza, although it is somewhat subtropical and there can be the capability of having summer outbreaks of influenza. And our last site is in Macha, Zambia, at the Macha Research Trust. This represents a very different population that we do surveillance on. It's sub-Saharan rural African with no influenza vaccination programs whatever, and this represents the Southern Hemisphere or tropical region for influenza virus spread. So these 3 sites feed into our pipeline, and we've spent 7 years really being very good at analyzing a number of different changes in seasonal influenza, some of which happen globally, some of which happen locally, and we've really worked our pipeline to very high efficiency in terms of isolating viruses and characterizing them. So of course, just as we were all prepared and ready to continue to work on influenza, in 2019, we of course had the appearance of COVID-19 or SARS-CoV-2. And everything I just described to you from influenza should be very familiar because multiple laboratories around the world started to do exactly the same type of things that we did with influenza, but we're just focused on SARS-CoV-2 and COVID-19. And of course, our center was nothing different. We quickly pivoted from being an influenza center to actually being a COVID-19 center of excellence. But essentially, all the components that I described for you were ones that we ended up putting in place so that we could do surveillance and monitor and characterize SARS-CoV-2 isolates. So to outline what I want to speak about today, it really falls into 4 areas. We're going to talk about my laboratory's ability to do SARS-CoV-2 virus isolations and how that work led to important observations that help determine the infectious virus period of an infected individual, as well as it helped determine effective timing of different tests as opposed to the nasal swab, antigen tests, as well as saliva tests. Well, I'll then talk a little bit about the ability of our laboratory to set up SARS-CoV-2 neutralizing antibody assays and how we help set up a pipeline to generate not only viruses to study, but also spike proteins that can be used for ELISAs and other serological assays. And for these, we applied them most effectively to determine which donor individuals might have plasma that was suitable for convalescent plasma studies, which are ongoing right now at the Johns Hopkins University, as well as many other institutions across the United States. And finally, we'll talk about something that's much more basic research oriented, our ability to isolate SARS-CoV-2 viruses, then led to our ability to characterize those viruses. And we utilize primary cell cultures derived from human respiratory tissues to characterize virus replication. And in this case, I'll show you some data that help us determine how variants can change their ability to replicate and spread within the respiratory tract using our model systems. So first, let's talk a little bit about SARS-CoV-2 isolation and how our work has helped understand the period in which a person is infectious, as well as how antigen and saliva testing can be applied most effectively to identify SARS-CoV-2 infected patients. So we early on set up a very close collaboration with my dear friend and colleague, Dr. Heba Mustafa, who runs the clinical diagnostic virology here at Johns Hopkins Hospital. And she very early on not only established in-house diagnostic techniques that were qPCR based, but also set up a SARS-CoV-2 sequencing pipeline so that virtually every sample that comes through Johns Hopkins University is tested by qPCR in her laboratory, and then approximately 50% of those samples are moved into her virus sequencing pipeline. So early on, we partnered with her to really investigate what was a rather confusing question with early qPCR testing, and that was individuals could actually test positive initially, but then could test positive by qPCR for many days afterwards, sometimes up to 19 to 23 days. Oftentimes these were low-level qPCR tests, but early in the pandemic we had a very difficult time understanding what that meant. Were these people truly infectious that whole time? Was this just some small leftover virus that was present within the individual? So Heba worked together with my laboratory, and we performed virus isolation on samples from individuals who were testing PCR positive for SARS-CoV-2 for long periods of time after their initial test. And in this table, yellow values represent samples that we tried to grow virus from, which we were unsuccessful at. And the orange samples represent samples where we were able to grow infectious virus from these individuals. So what you can see is most individuals did not have any infectious virus associated with these long-term PCR positive samples. But there were a few individuals that were able to isolate virus, sometimes as long as 22 days after their initial positive test. But when we analyzed the demographics and the clinical characteristics of these patients, we realized in all of those cases, people were showing symptoms for a much longer period of time than average. So individuals who were able to have infectious virus cultured from them 14 or 22 days post-infection were also showing much more symptoms at those times. So we quickly realized that most long-term PCR positive test results do not correlate with the presence of infectious virus, meaning that those individuals were probably not capable of shedding virus and transmitting virus. But if an individual was symptomatic, then there was a higher likelihood that they could shed infectious virus for longer periods of time. And that helped our hospital to very carefully institute quarantine rules and isolation rules, particularly for their staff, that allowed individuals to come back to work at a much faster rate and kept our hospital functioning over time. Now, that was from relatively healthy individuals in that population. But soon after that, we started to realize that individuals who were immunocompromised were actually showing a very different disease potential. In immunocompromised individuals, their infectious period, their symptomatic period was much, much longer. And in fact, many hospitals were seeing people who were on various cancer therapies or immunosuppression therapies who were showing symptoms for 60, 90, 120 days after their initial diagnosis of SARS-CoV-2. So in this study, we partnered with Jennifer Bard at the University of Southern California to identify some pediatric patients who were undergoing therapy for lymphomas in which they were being severely immunocompromised. And in this case, they identified 3 individuals who were actually shedding virus for a relatively long period of time after their initial diagnosis. And I'll focus on one of those patients just to give you an example of what we found there. So in this individual, patient 3, they were initially positive for both PCR, for subgenomic RNA, which Dr. Lai just explained is the replicating forms of RNA detected in infected cells, and also for infectious virus by tissue culture. But you can see that this person continued to be PCR positive for genomic as well as subgenomic RNA up to 162 days after that first diagnostic test for SARS-CoV-2. And importantly, at 139 days post first infection, we were able to isolate infectious virus from that person's nasal swab. And when we were able to sequence that virus, we saw something very, very unique. And that's shown here in this numbering table here. In this table, dark blue colors represent mutations that penetrate at 100%. And as the color gets lighter, it represents mutations that penetrate to a smaller and smaller percent of the overall sequence. But if you compare the sample at 139 days post-infection, you can see that there are a number of mutations that this virus has that is identical to the virus that the person was first infected with. And in fact, the genetic clade of this virus is exactly the same as the clade of the virus that was present at day 1, indicating that this is really virus that was probably still in this person's system from the first day of infection. But you can see by the accumulation of other dark blue signals here that that virus in this individual over the course of 139 days had acquired a number of novel mutations, both in the polymerase genes here, but importantly, also in the region of the spike protein, including several amino acid changes that are close to antibody binding site and may represent escape from neutralizing antibodies. So what this study showed was that in immunocompromised individuals, one could actually have very, very long-term shedding of not just RNA, but of infectious virus. And that virus would continue to evolve in those patients, accumulating mutations across the entire genome. And so we're currently isolating these viruses and we're studying these viruses to see how their viral fitness, their replication, compares to that initial virus this person was infected with, to see how likely it is that these immunocompromised individuals might be capable of evolving viruses that are better able to replicate in humans and perhaps spread from person to person. So having understood a little bit about infectious virus and how it relates to PCR in individuals and how that relates to their symptomatic disease, and their immunocompromised status. We then started to work with a number of laboratories to optimize viral testing, to try to move away from the nasal swab test, PCR testing, to try to get tests that were both faster as well as perhaps less invasive and easier for individuals to take rather than a nasopharyngeal swab. So our first work really was with Charles Cooper, who is at Becton Dickinson, and with him and my great friend and colleague Yuka Manabe, who is again part of the diagnostic group here at Johns Hopkins Hospital, we started to compare the ability of standard nasal swab mediated qPCR tests and antigen tests, which detect the viral proteins that are present in an infected test. And we wanted to see how those correlate with infectious virus. And in this publication, the first thing I wanted to say is that very early on, my laboratory started to use a subclone of Vero cells called Vero E6 Tempris 2 cells. These were a kind gift by an old friend of mine, Makoto Takeda, from the National Health Institute in Japan. And these cells overexpress a protease, Tempris 2, which is important for SARS-CoV-2 infection because it activates the spike protein and makes it better at mediating fusion between the viral envelope and the cellular envelope. And so what we did was we took a whole panel of samples from individuals at various stages of infection and tested them by qPCR as well as by the antigen test and tried to correlate that with infectious virus. And while the RT-PCR test on the nasal swab was the one that was obviously the one that was earliest in terms of showing a signal and highest in terms of its specificity and sensitivity, the important thing about the antigen test is that the antigen test here in yellow overlapped very significantly with the presence of infectious virus in the nasal swab sample that was being analyzed, suggesting to us that the antigen test, while not as sensitive as the RT-PCR test, was perhaps much more better at recognizing the stage of infection where infectious virus was being made by an individual, and therefore they're more likely to be spreading the virus at this time. And again, our cell culture system was an advancement over regular Vero E6 cells in that the cells were more susceptible to infection, and therefore we were able to detect lower amounts of virus in these nasal swab samples and find this correlation between the antigen test and the infectious virus. And again, this is important because antigen tests are cheaper than qPCR tests, and they're also able to be done within a matter of minutes. And so antigen tests represent that testing that can be done on site on an individual with results coming through relatively quickly, which could tell you if that person is actually infectious or capable of shedding virus. After completing that study, we actually partnered with Christopher Brook, who is a faculty member at the University of Illinois here in the United States. And interestingly, Christopher was an undergraduate in my laboratory about 15 years ago, and he went on to do a fantastic Ph.D. He did his postdoctoral work, established himself as an independent investor at the University of Illinois, and then decided to come back to me and ask me to work for him in a larger study that he was putting together. And his study was very unique because at the University of Illinois, they were testing students, faculty, and staff 3 times a week with a saliva test. And if you tested positive, you could go into quarantine, and in quarantine, you were asked to provide every day a sample, a nasopharyngeal sample, as well as a saliva sample, so that we could follow the course of infection and see how various tests could detect the presence of SARS-CoV-2 in your samples, and how that correlated with, again, the presence of infectious virus. So here are just 4 examples of up to 60 individuals that we studied for 14 consecutive days after their first positive SARS-CoV-2 test. And in This aqua color is the saliva qPCR test that Chris's lab developed. In blue is the nasal qPCR, the gold standard for SARS-CoV-2 diagnostics. The red X's are samples that are viral culture positive. And in this gold color is the time period in which an antigen test was positive for these individuals. So you can see we have 4 different measures looking for SARS-CoV-2, and we follow these individuals for up to 14 days after their first positive test. So the important thing to see here is that the saliva test and the nasal PCR test are actually fairly good in terms of detecting viral RNA. The saliva test is oftentimes a little bit lower in terms of its sensitivity. The CT values are a little bit higher than the nasal swab. But essentially, the entire period of time that someone is nasal swab positive, they're also saliva positive. You also see here the correlation where the antigen test is most often positive when infectious virus is present in these samples. So you can see that in this individual, and in fact, in this individual as well here too. So again, confirming the fact that antigen tests could be useful for detecting people during their infectious period of time. And the other thing that was interesting here was that the saliva test was always lower than the nasal swab when infectious virus was present in the sample. But when the saliva sample started to show a lower CT value than the nasal swab sample, invariably that person was not positive for infectious virus. So this told us a few things. Using a sample that is not as invasive as a nasal swab, simply collecting saliva from an individual, you could see the period of time in which they were shedding RNA and perhaps even infer their relative infectiousness based on the CT value of that saliva sample. And Chris and a large other group of individuals have come together to further characterize these individuals and to get a much deeper understanding of the kinetics of virus production, infectious virus production, and host responses to infection in this and a couple of other papers. So I hope I've shown you that our ability to really fine-tune virus isolation using influenza translated almost immediately to SARS-CoV-2 and was able to help provide some important insights into a person's infectious period, as well as to the ability of various tests for SARS-CoV-2 to show their correlation with when a person could be shedding infectious virus. I'll next turn and talk a bit about our work on serology, and in particular my laboratory's work on SARS-CoV-2 neutralizing antibody assays. And here we applied this specifically to the work here at Johns Hopkins University that was designed to identify donors for convalescent plasma transfer studies to treat individuals that were infected with SARS-CoV-2. So because my lab was getting quite good at doing virus isolation, we again paired with Dr. Mustafa to try to get a much more broader understanding of the viruses that were circulating throughout the Baltimore, Washington, D.C. area. And we collected, here's just an example of about 55 viruses. We actually have about 65 different variants of concern and variants in general that represent unique mutations in the SARS-CoV-2 genome. We have grown all of these viruses up. We have sequenced our viruses, and most of the time the viruses that we grow up are almost identical to the nasal swab that they were derived in, because we use these Vero Tempris 2 cells from Makoto Takeda that allow the virus to really be maintained in its initial state. So this has given us a panel of viruses which we can perform virus neutralization assays with. But what it can also do is it provides us sequences for us to make the antigens that we could do things like ELISAs for to measure general serological responses. And importantly, we can generate spike protein from exactly the same strain that we want to do our neutralization assay for, and therefore get a much more accurate reading of total antibody binding by S-ELISA and neutralizing activity assay because, again, the spike protein sequence of our ELISA antigen and our virus for neutralization is identical. So we worked with Mike Beatenbaugh's laboratory, who is a protein expression guru, who was able to take small-scale transfections of plasmids expressing both the spike protein and the SRBD, scale up those cultures, use large-scale protein purifications, and allowed us to then incorporate some high-throughput ELISA steps to move this process through so that we can literally analyze hundreds of samples in any given week for their antibody concentrations. And again, the first thing that we did was we tried to characterize a panel of individuals who had recovered from SARS-CoV-2 to assess their antibody responses and to try to understand if there was a particular population that might be the source of the best high quality antibodies in their plasma for use in the convalescent plasma donor procedure. So what we did in collaboration with my great friend and colleague Sabra Klein here at the Johns Hopkins University School of Public Health is we performed a couple of different kinds of ELISAs, a commercial ELISA, an ELISA measuring the total antibodies to the spike protein, and an ELISA specific for the receptor binding domain of spike. And we combined that with neutralizing antibody data that my lab generated to generate this scoring system where individuals were ranked based on how high their antibody signal was for these 4 separate assays. And together we then were able to select out from this large group of individuals about 12 individuals that we thought had the strongest and the most broadest antibody response. We then analyzed the clinical and demographic factors with these individuals and were able to show that there were several factors. The age of the individual, the older the individual, the stronger the antibody responses were. But that was intimately linked with sex, meaning that the males were the ones that were providing the highest titer, and also with hospitalization, meaning that disease severity was also equivalent to higher amounts of antibodies at various times post-infection. So this allowed us initially to focus on individuals that could be the source of high quality antibodies and then simply use the commercially available test to confirm how high these individuals were to try to select out the best plasma for use in the convalescent plasma studies. And I'll point out one thing that's very popular to talk about now, antibody waning. And we were able to show that even in this early convalescent plasma study, these are days since their positive swab, you can see that antibody waning was occurring early on after infection, and even within 60 days, you're seeing a general decrease in antibody levels after infection. So this concept of antibody waning does occur in infected individuals, as well as we're now seeing it in vaccinated individuals. But again, our studies here were able to help us sort out the best potential plasma donors to get those plasma into our study as quickly and efficiently as possible. We then followed up this study by working with Margie Ackerman at Dartmouth University, who has a very interesting way of analyzing antibody functions that are unrelated to neutralization. And she does this by looking at the Fc region of antibodies, as well as the subtype of antibodies. And she can quantify the amount of these antibodies that are present in the serum sample, and also quantify how much of those antibodies are recognizing SARS-CoV-2 antigens. And in this way, she could set up a much more broader scale antibody analysis that takes into account the things like antibody opsonization, complement fixation, Fc-mediated killing of cells, and can incorporate those aspects of the antibody response together with our neutralizing antibody response. And in this complex bioinformatics system that they were actually able to sort out 4 different types of antibody responses in individuals who had been infected with SARS-CoV-2. And most importantly, this purple group, group 4, was a group that had the highest neutralizing antibody titers, but also had the highest titers of antibody that would mediate these other Fc-mediated functions of antibody. So clearly we could find individuals who were generating a very, very strong and very, very broad antibody response after infection. And we're continuing to study these individuals to see what it is about their immune systems and responses is stimulating such a broad polyfunctional antibody response to compare to other groups of individuals. Okay, so in our SARS-CoV-2 neutralizing antibody study, we really went deep in terms of understanding more than just neutralizing antibodies to try to get a better understanding of the overall response to virus infection that was generated in convalescent plasma individuals. I'm now going to turn to some of our studies of SARS-CoV-2 in our primary cell culture systems, which are really aimed at directly assessing how variants are changing their ability to replicate and spread, giving us insights into what variants might be of highest concern to humans. So with that, I'll again come back to my collaborators, Heba Mustafa and Paul Morris. They are constantly sequencing viruses at Johns Hopkins, and this is just a picture of sequences from March of 2020 through September of 2021. You see the scale here, and basically the different colors represent different variants that have been dominant in our population. I'll focus the most on this part of the graph, where the purple is the alpha variant that Dr. Lai introduced previously, and in brown is the delta variant, which has spread not just in the US but globally and is shown to be a more transmissible virus and also able to cause more disease in many populations. So you can see that here at Johns Hopkins, the beginning of 2021, we had a large number of alpha variant cases, but that quickly transitioned over to almost exclusively delta variant in our population. And to this day, we're now isolating essentially only delta variant in our sequencing efforts. So this represented to us a great ability for us to now compare delta variant infections to alpha variant infections, because we had such a clear delineation of times when alpha variant was dominant versus delta variant being dominant. So we took samples and characterized them first for their overall CT value to look at their viral load. And we have here three comparison groups. A pre-alpha group, which consisted of viruses of a lineage B1.2, the alpha infected individuals, and the delta infected individuals. And what should be clear here is that the delta individuals all had on average a lower CT value, meaning more viral RNA when compared to alpha virus. Curiously, this was very similar to what we saw early in the pandemic, before vaccinations were being used, in that the earlier B1 viruses also had a lower CT value and therefore a higher viral RNA load compared to alpha. And again, we think this is because during the time that alpha became dominant, we were also initiating our vaccination efforts. And so some of this may be reflective of individuals who were vaccinated against alpha and therefore might have lower viral loads. So we broke up these samples into exactly that, the vaccinated versus the unvaccinated groups. And here we're again showing you the CT values of their qPCR results. And the important result here comes when we compare the individuals who were vaccinated but then infected with alpha to the individuals that were vaccinated and then infected with delta. And you can see that there's a much lower CT value associated with individuals who were infected with delta but were vaccinated when compared to alpha, indicating that this lower load is not just present in the unvaccinated population, but is also present in the vaccinated population, suggesting that delta could cause a much higher viral load even if you were vaccinated. So again, as I've talked about before, my laboratory does virus isolation. So we took as many of these samples as possible and performed virus isolation to ask for what the level of infectious virus was in these samples. And when we first looked at alpha versus delta, the summary is shown here in these pie charts. You can see that 30% of the positive samples were positive for infectious virus. In the alpha infected individuals. whereas the Delta infected individuals, that went up to 67% of the population. When it comes to when we split this up between the unvaccinated and the vaccinated individuals, you can see that the unvaccinated individuals, about 37% of the people were positive for infectious virus here. And with the Delta virus, that went up to 66% of that population. But what was also curious was in the vaccinated population, we also saw that a significantly higher percentage, up to 70% of the samples from Delta infected individuals were able to have infectious virus as compared to only 17% of vaccinated individuals compared with Alpha variant. And in this case, this definitely shows that the Delta variant is able to penetrate much more effectively in vaccinated populations as compared to the Alpha variant as it moved through the population. I think I heard my bell ring saying that my time is up. So I think I will stop here. And again, just emphasize the fact that our work on infectious virus has really allowed us to really get greater insights in terms of what it means for an individual to be infected and to actually point out their transmission period. And again, our work is also showing that Delta variant is much more dangerous than the Alpha variant in both unvaccinated as well as vaccinated individuals. And therefore, the work that is being put together to try to minimize Delta is certainly justified. And with that, again, I'd like to thank everybody for their attention, and I'm happy to take any questions if we have any time.
Thank you very much, Andy. Well, now we have a couple minutes for discussion. Let me remind you the discussion, you can use either Chinese or English. Well, I don't see any from the participants, so well, Xiru, are you there? I saw you.
Yes.
Okay. Would you like to add some of your data or experience to Andy's talk?
Andy, very nice talk. I enjoyed it a lot.
To him. That would be fine.
Yeah. Okay.
Hi, Shinru. How are you?
How are you? Very nice talk. I enjoy a lot. So it's amazing that you have a work to determine some antibody that are highly correlate to neutralization ability. And you mentioned that maybe it's FC mediated antibody. So do you have any idea the mechanism why some people just can produce more FC mediated antibody? So maybe after infection or vaccination, they got higher neutralizing antibodies.
Yeah, no, we don't know the details of that yet, but we are showing that those FC mediated functions enhance the activity of these convalescent plasma that's present, at least in vitro. And we believe that we have enough individuals who had donated convalescent plasma in that group 4 that we hope that in a few weeks, when we do all of our analysis of the convalescent plasma, we hope to see that signature that we would have hypothesized that individuals who got that plasma would have a much better outcome than individuals who got plasma that was less polyfunctional. So we're anxiously waiting to see our clinical trial results, which are going to come out probably sometime at the beginning of October, to see if that hypothesis is true. I think if we can show better protection with that polyfunctional plasma, then it really opens up a large area of research to understand what it is about those individuals that drives that broader response. But right now we don't know.
Yeah. Did you do any of antibody-mediated cytotoxicity test?
Yes, part of that was done in Margie Ackerman's laboratory to identify the individuals that had higher amounts of that.
Okay.
Yeah. So some of that was just looking at the FC regions, but some of it was also doing in vitro functional assays to mediate those types of activities.
Okay. Any other questions? Oh, Ken?
Yeah, it's a very interesting talk. I just have a brief question about your last slide. You said that unvaccinated patient has even higher infection rate than the unvaccinated.
Yes.
Does that mean that what kind of vaccine? And does that mean the vaccine does not have any effect on the Delta?
So most of our individuals were immunized with either the Pfizer or the Moderna vaccine. And what's important to note, and I failed to mention this clearly, is we're still seeing the protection of those vaccines against Delta. Because when we see our patients come in, 80 to 90% of the patients that are coming in are unvaccinated. And it's only about 10% of the patients that we're seeing are vaccinated. And here in the state of Maryland, we've reached a 70% of the population being vaccinated. So the vaccines are working. But I think what our data shows us that when you get a breakthrough infection, Delta can be as present as high as concentration in that person as it is in an unvaccinated person, which is telling us that Delta is a stronger virus. It's able to replicate, I think, more effectively than Alpha, and therefore can cause breakthrough infections that actually result in more amounts of infectious virus. So the vaccines are working, but Delta is banging that wall and making it more harder for those vaccines to really be as effective as they were earlier.
Well, Peter, you have the last question because we are a little bit over time. Peter, please.
I just want to make it short. I still want to think more about the last slide as a question by Professor Ken Wu. My interpretation, I might be wrong, is perhaps those vaccinated individuals who got breakthrough infections are due to highly infectious Delta variant. In other words, vaccine does work, and those less infectious virus will be protected. Therefore, the vaccinated who shows PCR positive and virus particle tend to have low CT value and high infectivity. That's my interpretation. I might be wrong.
Yeah, and I think the important thing about this too is that our comparison is limited by the fact that Delta infections occurred when many individuals were 5 to 6 months after their initial vaccination, whereas Alpha was occurring much earlier when people were really at their peak antibody titers. So we're unable to differentiate those two effects. But again, I do feel the vaccines are working against Delta. It's just that we're seeing more breakthrough infections than we did with Alpha. And I think it's a combination of Delta being a more aggressive virus and the vaccine-induced immunity dropping a little bit in the population.
Okay, thank you, Andy. And we are going to move on to the next session. So Andy, thank you very much to join us. And also, we really like to see you in the final discussion, okay? I think we are going to have very nice discussion.